Hello, today we're going to be talking about the cell. This is lecture 2 of Introduction to Biology. Over here, we have the types of cells, a eukaryotic and a prokaryotic cell. The difference between a eukaryotic and a prokaryotic cell is that a prokaryotic cell has no organelles, while a eukaryotic cell has organelles. Why organelles? Why do we have such specific compartmentalization in our cell? Well, the main function really is to store and contain different compartments. Because the cell has separate pH or concentrations for stuff we want to make, we need to compartmentalize. Com- we need to make compartments in the cell for specific stuff. For example, to digest and autophagy, we need a low pH. That's why we have lysosomes and digestive enzymes in those, which operate in acidic conditions or low pH regions. Then we also have membranes. Membranes are also an organelle, even though it might not seem. Membranes are actually sites for chemical reactions, and they have a unique combination of lipids, such as phospholipids, and proteins. Embedded enzymes and reaction centers also included, and chloroplast and mitochondria also have these membranes. So, as we can see, the cell has a lot of roles. Mainly, make proteins, make energy, and make more cells. And these are the three main processes that cells undergo. Building proteins. So proteins do all the work. For example, DNA. All of our proteins come from DNA to RNA to proteins, or the central dogma. So basically, all of our genes and everything, all of our genes and our expression is basically all dependent on proteins. Cells function. So we're going to talk about mainly about building proteins. So as I said before, reading DNA instructions basically what builds proteins. And the cell also not only builds or helps build, but also processes. So that includes folding, modifying, removing amino acids, adding other molecules. For example, glycolysation, which is in the endoplasmic reticulum, and it basically adds a sugar molecule onto amino acids. And it also makes glycoproteins, so basically carbohydrates with proteins for the cell membrane, because our cell membrane has glycoproteins. So organelles involved in this are the nucleus, the ribosome, the ER, and the Golgi and the vesicles, and this is the protein assembly line. This is very actually important because this is basically how proteins get made. It gets made in the nucleolus, specifically from ribosomes. Ribosomes get made in the nucleolus. Then from the ER basically helps create proteins and it ships it to the Golgi, which then directs it to sites like the membrane, the vesicles, maybe lysosomal proteins. So now we're going to talk about the nucleus's role, which protects DNA. And the nucleus is a structure. So the nucleus has a nuclear envelope, which is a double membrane, and it has pores. These pores basically regulate like mRNA transport and export, because all of our transcription will undergo in the nucleus. So when it wants to leave the nucleus, it'll go basically out of these nuclear pores. So basically, this is just what I talked about, mRNA export and import from the nuclear pores. Specifically, if you want to know, it's from the RAN-GTPase system that regulates mRNA export and import out of this nuclear membrane. So we're talking about the nucleolus, which is just not actually a separate organelle, but it's inside the nucleus. Basically, like I said before, its function is ribosome production, and it builds ribosome subunits from rRNA. I believe 5 rRNA are used to build a ribosome subunit, and they exit through nuclear pores. Ribosomes function. So after the nucleolus starts making these, its function is actually protein production. So it'll make proteins, like as we can see here. So these are ribosomes on top of the rough ER in this diagram. That's actually why we call it rough ER, because of the rough texture from the ribosomes. The smooth ER, which doesn't actually contain any ribosomes. So there's types of ribosomes. Free ribosomes. Free ribosomes are suspended in the cytosol, and they actually basically create metabolic proteins such as phosphofructokinase or hexofructose kinase, and such as these enzymes involved in like cellular respiration. And we have bound ribosomes, which basically make proteins for export, like cell membrane proteins. So now we're talking about the ER, the function of the ER, and this process of proteins, manufactures membranes, and synthesis and hydrolysis of many compounds. So basically, their main job is actually just to process proteins and make sure, like, all the proteins are fine. Any modification they need to add, just adding. Sometimes they need to add a sugar molecule onto the protein, or lots of other modification. That's basically what the ER does. Types of ER. So we have a rough ER and a smooth ER, and this is just like structural differences between them. Mainly, the smooth ER is more tubular, and the rough ER has just ribosomes on it. Smooth ER function is actually very different than rough ER. So they actually synthesize lipids, oils, phospholipids, steroids, and sex hormones, and they hydrolyze glycogen into glucose in the liver. And they also detoxify. It's a main important thing. So you would expect smooth ER to be found in a lot of cells in your gonads, or basically cells that produce a lot of sex hormones. The membrane factory. So now we're talking about basically the Golgi apparatus. So over here is a diagram of the Golgi, and basically by membrane factory, just saying like the process of after the ER, how the transmembrane glycoproteins enter here, and they join the Golgi apparatus, which basically just ships it out to membrane. Rough ER function. So the rough ER produces proteins for export out of the cell. Protein secreting cells included in their packaged in transport vesicles for export. Now, which cells would have a lot of rough ER? Which cells have a lot of rough ER? Well, it will be cells that actually have just basically need to produce lots and lots of protein. Synthesizing proteins. So over here let's see, we have a Golgi. So this is just basically after the Golgi, I guess, how it gets transported out. Because here's a signal sequence, and these signal sequences basically determine where it gets exported. So the Golgi apparatus, you could think of the Golgi as like a UPS truck. It basically tags ships and everything like out to everywhere through these signal sequences that we discussed before. So which cells would have a lot of Golgi? Basically, like we said, similar to the rough ER example, it will be cells primarily used toward shipping out proteins toward maybe the membrane or other cells for export. Okay, so over here, just vesicle transport. So this is just a diagram showing how a vesicle budding from the rough ER moves to the Golgi apparatus. So actually, more about the Golgi apparatus, since the slides don't explain it actually well. The cis side is going to be the receiving side. So over here we have a cis side, basically receiving it, and then the trans side will actually leave it. And basically, the cool thing about Golgi is they kind of reform and destroy themselves because as the vesicles join in, it'll form new cistern A, basically just tubular structures in the Golgi apparatus, and it'll destroy the vesicles start leaving from the trans end. So as you can see here, trans and cis, and this is the cistern A. That actually gets reformed and formed from the vesicles. So that's just putting it all together. This is the whole system, the endomembrane system, which includes the proteins being made and getting shipped. Now, this is just an overview of the cell, the eukaryotic cell. We discussed the endomembrane system, and now I think we're going to go more into deep about lysosomes and peroxisomes and mitochondria. And this is a plant cell. So if we look over here, the main difference between a plant and an animal cell as well, the plant cell has a way larger central vacuole, which is actually important for its structure. On top of that, they have a cell wall, which we don't. We only have a cell membrane, but they also have a cell membrane. So they just have another structural component toward it. Finally, the another major difference is actually animal cells will have centrioles, which are important in mitosis. So over here, as we can see, we have centrosomes and centrioles, but plant cells actually lack that. Making energy. So ATP is actually our primary adenine triphosphate, which is our primary form of like energy in the cell. Cells need power, so they take in food and digest it. They take in oxygen and they make ATP, and this is mainly done by the mitochondria. So lysosomes. Lysosomes are actually stomach of our cell. Like I discussed before, lysosomes just basically kind of digest the components in our cell that we really don't need. It cleans up broken down organelles or just waste in our cell. And this is through hydrolytic enzymes that are in the lysosomes that basically remove all the bad stuff. The important thing about lysosomes are actually only in animal cells. I forgot to discuss this before, but if you look at the previous pictures, plant cells actually don't have lysosomes. However, animal cells, we have lysosomes. So lysosomes fuse with food. This is just basically cellular digestion. I think I discussed this before. Autophagy. So basically, how lysosomes are actually a main component or a proponent of autophagy, as they actually conduct the whole thing. Their enzymes inside the lysosomes actually will break down whatever is needed. So lysosomes actually are one of the organelles in our body that work at a very acidic pH environment. So that's actually out of five. So how do organelles create a custom pH though? Because we know cytosol's around 7.4. Proteins in lysosomal membrane will pump hydrogen ions from cytosol into lysosome. So now as we know from just basic chemistry, an increase in hydrogen ions means more lower pH. And why do we need to do this? Well, enzymes are very sensitive to pH. They actually denature from pH changes and temperature changes. To make sure an enzyme doesn't denature, we need to make sure the enzyme's environment is suitable for its function. When things go bad. So basically, diseases of lysosomes are often fatal because, for example, what happens if you don't have lysosomes, there's nothing to digest anything. The digestive enzymes won't work, and we can't pick up any biomolecules. We can't even digest one. Okay, so there's a few storage diseases. Tay-Sachs. Tay-Sachs is a very common one, and these are from lipids. And then here's a few other ones, which really don't need memorizing. It's just good information. So, but sometimes cells need to die, and lysosomes can be used to kill cells when they're supposed to be destroyed. Some cells have to die for proper development in an organism. For example, apoptosis. Apoptosis is actually the autodestruct process in our cells. Lysosomes will break down and act as proteases and just kill the cell. So apoptosis. Apoptosis basically is programmed destruction of cells and multicellular organisms. So example of cells called uncontrollably, basically cancer, right? If P53 gets suppressed and our cell starts growing uncontrollably, all of our defense mechanisms that the cell has to basically stop uncontrollable cell growth, if it doesn't all work, then apoptosis will be initiated, which is through many stuff. For example, cytochrome C in our mitochondrial membrane basically is linked to apoptosis. So a lot of pathways and chains for apoptosis to occur. So cells must convert incoming energy to forms that they use for work. So ATP is the form we use, but we ingest glucose, right? So we convert glucose to ATP and sunlight to ATP in plants, chloroplast. And we'll talk about these two, like anabolism and catabolism, like basically these two processes later. So now we're talking about mitochondria and the chloroplast. So they both are involved in energy processes in the cell, but the chloroplast is obviously just not found in animal cells because we don't do photosynthesis. Due to the endosymbiotic symbiote theory, that we both have double membranes because they were actually hypothesized to actually be an organism itself, but then it just got digested. So they have double membranes, and they actually have their own ribosomes and DNA. They kind of just like function by themselves, isolated from the cell, but they're still part of our cell. They just kind of do their own thing. This is the function of mitochondria. Main function is cellular respiration to generate ATP. Basically, from the breakdown of sugar, fats, and fuels, we will produce around 38 ATP, and this is in the presence of oxygen. So, mitochondria has a very different structure. We have an inner membrane and outer membrane, basically because there's two membranes in the cell. And we have cristae, which are folds in the mitochondria and a matrix. The point of the cristae is actually to increase the surface area, because the more folds we have, we'll have more surface area, which is better for the cell for energy processes. So this is just cycle, cellular respiration, ETC, specifically electron transport chain. We'll discuss more about this later, but this is just basically like an overview of like how we produce all the ATP from oxidative phosphorylation. Okay, so this is just basically how the mitochondria divides. So basically, as we can see, it divides like basically like binary fission, and basically just tells us that evolution of eukaryotes, they probably came down from a long line of like archaea and prokaryotes. So basically, just an overview. All like eukaryotic cells have mitochondria, but for example, cells that have a lot of mitochondria are probably cells primarily used to like action for actions, movement. Like for example, muscles in your leg, basically need, or stuff that would actually need energy, a lot of energy. So like muscle cells, for example, that are used to move around, and nerve cells, which actually just need to produce a lot of energy. So these are chloroplasts, and we have a few different types. An amyloplast, which just store starch and roots and tubers. Chromoplast store pigments, and chloroplast, which store chlorophyll, the pigment, and function in photosynthesis. Over here we have structure. We have thylakoids. Thylakoids are actually the main thing that like absorbs it. And we have a stroma, which is the aqueous space, kind of like the cytosol. And we have a granum. Granum is just a stack of thylakoids. This is the electron transport chain of in photosynthesis. As we can see, it's actually pretty different. We have a photostem 2 and photostem 1. We'll also discuss more about this in depth later. Uh, so chloroplast. So basically, as we talked about, they function photosynthesis, just generate ATP and synthesize sugars. That's the main job is actually to synthesize sugars. And okay, why are chloroplasts green? Basically, this is just due to the pigment chlorophyll, and it's because basically chlorophyll will absorb red and violet and blue light and actually reflect green light. So a common misconception actually is that chlorophyll's green or something like that, but it actually reflects the color green off of it. Instead, it mainly absorbs, I think, red light, and also blue and violet pretty high as well. There's just some differences over here, as you can see, from mitochondria and chloroplast. This is the symbiote theory, which I kind of briefly explained, I guess. Mitochondrial chloroplasts once free living bacteria, and they were engulfed by an ancestral eukaryote. That's why we have, like, they have their own DNA, and they kind of function differently, I guess, and they have a double membrane. So this is kind of just shows how everything kind of comes together in energy. So from photosynthesis to creating glucose, we'll then break this down and create and for respiration and just release CO2 and H2O and how that's also back then used to for photosynthesis. So just everything's connected basically. So how do we store food and water? So food and water is mainly stored as stuff called vacuoles. So for example, plant will have a central vacuole, which is way bigger, and animals cells will have vacuoles, but we could also there's also something called a contractile vacuole, and there's also food vacuoles. So a food vacuole is associated with stuff like functions such as phagocytosis, which fuses with the lysosomes. Contractile vacuoles basically pump excess H2O. It's basically when the like isomodic environments like or basically it's hypotonic, so it wants to pump out H2O because there's too much solute in the cell attracting water inside, so they want to pump out H2O. So there's just a bunch of functions for vacuoles and plants, storing just basically storing pigments, compounds. Now I go about peroxisome. Peroxisomes is I think an important thing function I'll list in the slide is beta oxidation, and it includes breaking down basically fatty acids and just basically producing acetyl CoA, which is used in like a metabolic process we're gonna discuss about later. Making new cells. So basically, I think the last thing we're gonna talk about is cytoskeletons. And cytoskeletons basically function by structural support other than the cell membrane, maintaining the shape and providing anchorage. Cytoskeleton actually includes many protein fibers, which include microfilaments, intermediate filaments, and microtubules. So over here is just a basic the function of the cytoskeleton. This is very much worth memorizing all these three. It's very important to know the differences. I'll just go over an overview. Microtubules basically include tubulin, and they have an alpha and beta tubulin dimer. Uh, Basically just a dimer consisting of an alpha and a beta. Maintenance of shell shape is the main thing. They include cell motility, as cilia and flagella are made of tubulin. Chromosome movement and organelle movements all changed by microtubules, which is just tubulin. Microfilaments, which include actin. Actin is main thing is muscle contraction, very important. Maintenance of cell shape from the cytoskeleton, and cytoplasmic streaming is also pretty important. Finally, intermediate filaments, which include like keratin, like proteins like that. They include anchorage of nucleus and basically the formation of the nuclear lamina. Know that microtubule is actually like the widest and the biggest, while microfilaments are the smallest and intermediate are the middle. Here's just a picture of a cytoskeleton. Centrioles over here. Basically, as I discussed about before, centrioles, which are actually only included in animal cells, are important for cell division. So the difference between centrioles, centrosomes is kind of like the area, I guess. Centrioles are actually the microtubule pairs, and basically they organize the microtubules and the spindle fibers later using mitosis, which we'll talk about later. So we're talking about some junctions, I guess, which is actually going to be the last thing we're going to talk about. So basically, plasmodesmata are channels, and that's basically how stuff will go through plant cell walls, like solutes such as RNA and proteins will pass. And this is through plasmodesmata, which is pretty fast and passes cell to cell. It's similar to something called gap junctions, which are in animal cells. Gap junctions basically are the fastest type of channel, and they move it through adjacent cells. We have desmosomes, which function as they have rivets, and they actually fasten cells together into strong sheets. I think they use intermediate filaments to connect, and tight junction, which are mainly for like liquid protection and fluid leakage. So basically, it prevents leakage of extracellular fluid. We have a picture over here. It's common, as we can see, intermediate filaments, mainly involved with desmosomes, tight junctions up top, and gap junctions. So over here, just the extracellular matrix of an animal cells, which is basically includes cytoskeleton. The ECM made up just a bunch of mainly glycoproteins, and collagen is the main protein involved in the ECM. And basically over here we have collagen fibers, and the collagen fibers are winded by a proteoglycan complex. And fibronectin is just a main thing that's basically connecting from these integrins or on top of the integrins. So that's all for today. This is just a few overviews of basically what we just talked about. And that is all for the tour of the cell. I'll see you next time.